So today our seminar speaker is our very own GGI department member, Dr. David Ashbrook. David received his PhD in systems biology from the University of Manchester and completed postdoctoral research on the epigenetics of Gulf War illness at the University of Toronto and here at UT working on sequencing and behavioral genetics projects in rodent genetic populations. In 2020, David joined the GGI department as an assistant professor, where he has taken the lead on a number of projects, including deep sequencing, telomere to telomere sequencing, and pangenome assembly of the BXD recombinant inbred mouse family, one of my favorite mouse families. We have Thanksgiving every year. He is currently leading a team researching the impact of genetics and diet on longevity, and is also investigating the impact of genetic background on familial Alzheimer's disease. David is an integral member of our department, is highly collaborative, and an expert on systems genetics, G/E interactions, and omics integration in diverse rodent genetic models. He has over 22 peer-reviewed publications, including several in high-impact journals. He is currently supported as a co-investigator on 3 NIH U01 or R01s, and hopefully funded soon as PI on his own new R01 award, but it is a reward after all. So Dr. Ashbrook, welcome, and thank you for sharing your latest research with us today.
Thank you for the very kind introduction, Megan. As Megan said, I'm David, I'm an assistant professor here at UTHSC, and I'm going to talk to you about Thylal Crosses, artificial intelligence, and mouse models of Alzheimer's disease. But what I'm really going to talk to you about is complexity. All biology is complex. We have millions of variants interacting with every part of our environment, whether that's what we eat, the people that are around us, where we grew up, our age, our sex, and that affects gene and protein function and expression at the cellular level, at the tissue level, at the whole organism physiology, and that leads to behavior. And all of these things work together. So even something that we think of as really simple, so an early onset familial Alzheimer's disease, you can find the exact variant in a family that causes early onset Alzheimer's in that family. You can have decades of difference in age of onset. So you can have huge differences in the physical and cognitive symptoms they show, and we don't understand why that is. And we have a tendency to be reductionist and sort of try and ignore complexity, but really we need to understand complexity, to embrace it and understand all those connections to be able to get a real understanding of real biology and real disease. And we need experimental systems and new technologies that allow us to actually look at that complexity. Because if I gave you a genome of a mouse you've never seen before, tell you what its environment has been in, and then ask you, how long is that mouse going to live? Is it going to have insulin resistance? Most of the time, you're not going to be able to tell me. And if we can't do that in a model organism that we know so much about and have been able to control the environment so well, how on earth are we expected to do this in humans? And that's really what we want to do in precision medicine, is say, me as an individual, I know my genome, I know my environment, how long am I going to live? Am I going to develop Alzheimer's disease? So we have many ways to look at this complexity, and I'm going to talk to you about a few of these. I'm going to talk to you about Thylal Crosses as a way to increase genetic complexity and interrogate that genetic complexity in a controlled and replicable way. I'm going to talk to you about the Alzheimer's disease BXD mouse model that we're using, which is a sort of specific Thylal cross, and some of the background of that. I'm going to talk to you about artificial intelligence to get at behavior. Some people may argue that it's not artificial intelligence, it's neural networks and machine learning, but I'll leave that argument for another day. As a way to get sort of more complex data from the kind of behavioral tests and cognitive tests we do. And then I'm going to talk to you about a large brain imaging project we have and some of the deep phenotypes and new phenotypes we're able to gather from that. So first things is a Diallel cross, and it's a simple thing that has been used for decades, and it's simply all the F1 crosses between a set of inbred strains. And when we do sort of our experiments in mice, often this is done in C57 black 6J. So you'll see studies, papers saying, we did this in a wild type mouse. And what they mean is they did it in C57 black 6J, which is a single genome. It's like doing every single experiment in human biology in me and assuming that it's going to generalize to every other human being on the planet. And we know that C57 black 6J is very different even from C57 black 6N, which is a sister strain, very few variants between, but there's differences in GABA receptors that Megan works on, and therefore in their response to drugs of abuse, in their response to Tylenol, paracetamol, the common painkiller, in their response to many other phenotypes, behavioral phenotypes, disease phenotypes that are really important for us as humans, and yet we assume that these represent not just all of mousedom, but all of mammalian species. So what can we do? Well, first thing we can do is look at other inbred strains. We have hundreds of inbred strains, 50 or more of them have been fully and deeply sequenced. And now we can look across many strains and say, does our perturbation, does our experiment generalize to different strains? And if it generalizes across these many varied mouse strains, then we can start to say, okay, this generalizes across mousedom. We can then be more confident in saying this generalizes across mammals and therefore is applicable to humans. And I think this has been sort of one of the big problems in sort of systems biology, well, in all biology, is that we sort of just do things in a single strain and don't include genetic diversity. So how about Thylal crosses? Well, they've been used for many decades, and we'll just sort of look in on this simple Diallel cross here, which is between C57 black 6J and DBA2J. And we know the entire genome of both of these F1s because they'll get one copy of each chromosome from the black 6 and one copy from the D2. And this can tell you something about phenotypes. So you can tell if a phenotype is additive because the two F1s will be approximately halfway between the phenotype of the two parents. You can tell if a phenotype is dominant. So for example, if we said the C57 black 6 phenotype is dominant, so for example, coat color, we'd expect both of these to be black 6-like, so have black fur. We can also tell things about parent of origin effects. So perhaps it depends which parent you got a gene from. So let's say this is the mother, and you have a maternal parent of origin effect, this F1 will be like the black 6, and this F1 would be like the D2. And this can tell us about varieties, so variation between populations, and a bit about the phenotype. And we can apply this to genetically engineered mouse models as well, so GEMs. So again, we have the same issue with knockouts and knock-ins and transgenics, that they're usually done on a single genetic background, usually some form of C57 black, sometimes 6J, sometimes 6N, sometimes even the people doing the experiment don't know which. And we have this same issue that everything's done on the same genetic background. And we know genetic background can have a huge effect. So a good example of this, it was a series of experiment done by our collaborator, A. Palmer, in his lab, where they crossed knowns of knockouts to different strains and looked at the phenotype in those strains. And what they saw for exploration, for example, one of the knockouts on the background, exploration was really reduced. So if you'd just done that single strain, you'd say, aha, this gene is to do with exploration and activity. In another strain, there was no difference at all to the non-transgenic. If you'd done that single strain, you'd have said, okay, this gene has nothing to do with activity, let's move on. And then think about that and how scary that is. How many times do we look at a list of genes that we're interested in and go, okay, the knockout had nothing to do with my phenotype. There was no change in my phenotype. Hey, they even looked at my exact phenotype I was interested in. I can not ignore that gene. It's nothing to do with what I'm doing. And it purely depended on which genetic background they chose to do that on. So my first sort of ask of this talk is please add some genetic variation into your studies and please think about genetic variation. But none of this tells us about what variants are causing this difference. They're just saying there is a difference. And to look at that, we can use recombinant inbred strains. And I probably don't need to tell anyone that's on this talk about RIs, but I'm going to tell you a little bit about them anyway. So these are sets of strains. Each member of the family is fully inbred, and they're all derived from the same parents. Sometimes that's 2 parents, like the BXD. Sometimes that's 8 parents, like the CC. But each of these lines is now fully inbred and a unique mosaic of B6 and D2 alleles in the case of BXD. And because they're fully inbred, they're homozygous at each position. And these have been used, again, for decades to map out QTL, so regions of the genome linked to a phenotype of interest. But one limitation of these is the number of them. Even for the BXD, which is the largest and most deeply phenotyped family, we only have about 140 extant members of the family. For something like the collaborative COS, there's about 60 members of the family, and we would love to have more. And one way to do this is to use a Diallel cross. So here we can imagine 140 BXDs by 140 BXDs, and each of the F1s, because they've come from inbred parents, is isogenic. We know exactly what the genome is going to be. We've sequenced the BXD deeply, so we know what the genome is going to look like. And they are replicable. Because they're isogenic, we can use as many of these across different labs, across time, in different places, at different ages. We can take out the brain in a genetically and environmentally identical individual. And yet we have 200,000 of these we can use. And that gives us power to look at things that are difficult to look at, like epistasis, gene-gene interactions, where you need a large number of strains to get the power to investigate them. It allows us also to do prediction. We have tens of thousands of phenotypes, including expression and protein expression in the inbreds, in the BXD. And now I want to say, okay, let's take a group of these F1s that we've never seen before. We know what their genome is, but we've never phenotyped that genome before, and make a prediction. So this allows us to take that sort of first tentative step to precision medicine of saying, here is a genome and environment you've not seen before. Let's predict what the phenotype is going to be. It also gets us around some of the complaints that have been made about inbred strains, that they are homozygous at every loci. Well, in these F1s, they're going to be heterozygous at many of the loci. So that's Thylal Crosses. And we can do the same thing with the GEMs, with the genetically engineered mice. We can cross them to recombinant inbreds. And this means we can now map those modifier alleles. So why, when you have it on one genetic background, does it increase activity, and on another genetic background, does it decrease activity? And we can map those QTL. And this is basically what we've done with the AD-BXD. So this is the Alzheimer's disease BXD model, and they're based on the 5xFAD transgenic mice. So these were developed about 15 years ago, and they include the human amyloid precursor protein and 3 known familial Alzheimer's disease mutations in that, and the human presenilin gene and 2 known familial Alzheimer's disease mutations in that. And they have a really strong Alzheimer's-like phenotype. And this humanized transgene is expressed neuron specifically with Thy1 promoter. And this is then been crossed to the BXDs. So this is this AD-BXD, Alzheimer's disease BXD model. You take heterozygous 5xFAD on the pure B6 background, cross it to the BXD, and now you have F1s, each of which you know the genome. It's going to have 1 chromosome from the BXD strain and 1 chromosome from the 5xFAD parent. But within each litter, half of the litter will have the transgene and half of the litter will not have the transgene. And this means you have a perfect control. These 2 individuals are genomically identical, except for the transgene. They've come from the same mother, so they've had the same maternal environment. They can be raised in the same cage together. So they're environmentally fantastic controls, and therefore any differences must be due to the transgene. When you look across the population, you can then map where in the genome causes these differences. And this is sort of work that was done by Catherine Kraszowski and her lab, first here at UTHSC, and then moving up to JAX. And this ADBXD replicates many of the features of Alzheimer's disease, not just the cognitive features, but sort of physical features. So decline before you have physical decline, before you have cognitive decline. And there's remarkable variation between the ADBXD. So we can look up here at amyloid beta levels. And if you just cross the 5X FAD to the B6 or D2 parents, BXD population, you get roughly the same amount of amyloid beta. And yet when you look across the family, you've got something like the cross to the BXD14, where there is no detectable amyloid beta, or across the BXD75 that has almost double the amount of amyloid beta as the two parents. So you have massive variation in amyloid deposits. We can look at conditioned fear acquisition, or rather I should say Catherine did look at conditioned fear acquisition, and a low value is poor memory and a high value is good memory. And you can see massive variation across the ADBXD strains. What's even more interesting is because they're isogenic animals, you can take genetically and environmentally almost identical individuals at 2 different ages and look at the difference between 6 months of age and 14 months of age. And on the population level, you see a decrease in memory with age, which is what you expect in sort of both normal animals and in the ADBXD animals. But you can see a lot of variation within the strains. So most of the strains do what you expect. They have okay memory at 6 months, and then this declines with age. Some of the strains have fairly poor memory to start with and kind of keep the same. And then you have these that have good memory to start with and continue having good memory at 14 months of age. So 6 months of age is sort of early adulthood, 14 months is sort of late adulthood. And we kind of want to be these ones up here. We want to maintain our memory and we want to know the gene variants that are causing this. So because we've crossed them to BXD, we can do QTL mapping.
David,
so
this is Andy.
Yep.
Hey, David, I had a question and you didn't really, I might have missed it. So the one strain 14 had non-detectable levels of the A beta 1 42. And is A beta 142 expected to be a biomarker that correlates with the dementia phenotype? Because 14 also had really poor memory according, and so I'm trying to put those two together.
That is an excellent question. So in humans, we kind of think that amyloid beta has something to do with memory. And yet when you they've developed drugs that reduce amyloid beta, and they've had sort of no effect on the actual memory of patients with Alzheimer's disease. So this could be more evidence of that amyloid beta and the memory decline are actually separate from each other. Or it could just...
So the FDA obviously didn't talk to you before approving this latest drug.
They didn't talk to a lot of people, Andy. That's why it was so outraged. People who knew this has actually been old. This is old news now. This is probably 5 years old.
I'm sorry, David, but this is a really great way to maybe dissect out why those are discordant.
Yeah, exactly. And I will come back to that a bit in a second as well. So as I said, we can map these QTL. So this is amyloid beta at 6 months of age. And we have this QTL on chromosome 12. And this is the haplotypes up here. And we've ordered the strains by highest to lowest. And you can see that those with a high value have the green paternal allele, and those with the low value have the red maternal allele. So that's why we have the QTL here. And then we can look at something like performance on the Y maze at 14 months. So this is sort of a short-term memory task. And you can see this QTL on chromosome 4. You can see those with the high values have the maternal allele here. Those with a low value have the paternal allele. So this tells us two things. One, we can map QTL for using these ADBXD. And two, that different aspects of Alzheimer's-like phenotypes, so amyloid beta, short-term memory, mapped to different regions of the genome. So you have different regions of the genome modifying the effect of that transgene. And we can sort of split up these different interesting aspects of the disease using sort of this genetic model. So this is all work that was done by sort of Catherine and her lab. Now what we're doing is working on a large project using the AD-BXD and normal aging BXD as well, but I'm going to sort of ignore those. And we're just talking about sort of this here, where we're using the AD-BXD. We're using those 6 months and 14 months of age that Catherine and her lab used. We're looking at behaviors, and we're looking at deep brain imaging, both MRI and light sheet. And these are things I'm going to talk to you about for the rest of the talk. And this allows us to look at gene, by age, by sex interactions. We're using males and females, 2 different ages, and we're looking how different variants across the whole genome interact with this transgene. And we can do similar approaches for sort of any of your phenotypes of interest or diseases of interest. It's not just a brain thing, it's not just a behavior thing. People have worked on Huntington's disease, retinal phenotypes, and breast cancer. But I just sort of want to point that out and say we're concentrating on the AD-BXD here. So we're looking at a range of behaviors and sort of tests. So we've got sort of grip strength and rotor rod as measures of physical ability and various measures of cognitive ability. We've got the sort of their memory acquisition and Y-maze, which are the same tests that Catherine used. We're expanding the number of strains with some overlap. So hopefully these 2 data sets be usable together. We're using the snowflake maze, which is kind of an expansion on the Y-maze. It's testing the same kind of short-term memory. We're doing open field, which we use as both sort of a physical activity measure, just how much they move around, and sort of the normal anxiety type measures. And we're using the what, where, which test, which I will talk about in a second. And John is doing all these measures. So huge amount of animals, a lot of work doing that. And we're measuring all these cognitive measures here using Raspberry Pi cameras, and how Chen helped us set that up. So what's the what, where, which test? Let's see. It's basically asking, can you remember what object was where in which context? So we have 2 contexts. We have context 1, which has this sort of Lego sheet in the middle here, and context 2, which has sort of nothing on the floor, but has sort of red stripes on the wall. We put the mouse in here first with the sort of dog-shaped Lego model on the left and the tree-shaped Lego model on the right. Sits in there for 3 minutes, explores, learns which way around they are. The mouse is then put into context 2, where they're switched over. So the tree-like model is on the left, dog-like model is on the right. They're then taken out of those contexts into their home cage for a couple of minutes where they have to remember, and put back into this context 1. And now they have to remember, hey, this model is in the wrong place in this context. It was supposed to be on the left because we're in context 1, but now it's on the right. So the mouse that remembers about the context and the model will explore this one more because it's in the wrong place at the wrong time. So it's sort of a more complex memory task. And you'll notice these little spots on the mouse that are following it around, and these have been added by Deep Lab Cut. So this is a software package for pose estimation. Basically, you can define any point on the mouse and label those in a really small number of frames. So across all of your videos, you have to label maybe 50, a couple of 100 frames, and think about we're doing sort of 30 frames a second. This is a tiny percentage, but you get these really good, accurate and transferable models. So I can do this in a subset of videos as well, subset of frames, and it will transfer across all the frames within that video and any other videos we throw at it in a similar context. So this really reduces the amount of time and work you need to do for that. So we've got these 2 sort of tests of the Y-maze that I've got here, and you can see again sort of little spots. You can do all the normal things you do in a Y-maze, which is basically saying, does the mouse remember which arm it came out of last time, or does it go back into the same arm it went before? So going back in the same one, incorrect alteration. going back and exploring the one it hasn't explored yet is a correct alteration. But because we have all of these measures, that was an incorrect alteration there, we can look at anything else in here. So we can look at the amount they move, the speed which they move, are they stopping and grooming? We can measure sort of the amount of time they're grooming. Are they rearing up? So we can get those sort of activity sort of measures. We can see sort of if this one moves, fecal boli, which is another measure of anxiety. So we're doing the same sort of experiment that have been done for a long time, but now we're able to pull more and more information out of it. But Deep Lab Cut only sort of tells us the distance between these positions. It doesn't really tell us anything about behavior. So what we're using for that is SIMBA, which is simple behavioral analysis. And again, it's another kind of machine learning approach. You have the pose estimations and the videos. You can annotate a subset of those, and it will then learn and be able to put that across all the other frames on that video and any other videos you have. So this, again, it saves a huge amount of work of annotating every single video. And this SIMBA side is very much a work in progress. If there's any students interested in artificial intelligence behavior, please let me know. I'd be love for you to work on this. But it allows us to get at more complex things. So if we look at this what, where, which again, that's not, give me 1 second. Let's see if it'll let me select things. Okay, never mind. So if we look on the left, this is fairly clearly, it's exploring that one, and that's sort of exploratory. Whereas if we look here, it's just sort of using it as something to stand on, so it gets a bit of a better view. Now, if we're just using the distance measures, its nose is still pretty close to that object. So we'd say it's exploring it. But when we're actually able to look at the mouse, we can see it doesn't really care about the object. It's just using it as something to stand on. So this is something we'd be able to annotate in SIMBA. We'd be able to annotate that, yes, that's a sort of exploratory behavior, and that's just sort of being near the object. So again, this allows us to sort of get at more detailed phenotypes, and we can annotate anything else we liked in here. We could annotate the number of rears it does. We can annotate grooming. So again, it's pulling out more and more information from these seemingly simple tasks. So I've talked about the Diallel cross as a way to get sort of more genetic complexity. I've talked about SIMBA and Deep Lab Cut as a way to get at more behavioral complexity, but those are sort of two extremes of the spectrum. So genetics and behavior, what's happening in between? So one of the important things for behavior is the brain. So we can look at really deep analysis of the brain. So this is work we're doing with Al Johnson and his group at Duke. And we're doing this, he's doing this really, really deep MRI. So we've got a spatial resolution of about 0.015 millimeters cubed. And to give you some sort of idea, that's almost 300,000 times the resolution of a normal human MRI. He does this by a number of methods. There's both sort of technological sort of method development. There is analysis and bioinformatics technical development, and also the amount of time we can put a mouse in there. So this is euthanized mice. So we can put them in there for 12 hours or 48 hours, depending on the resolution we want. And that's just not possible in humans. So what we do is we euthanize the mice here at UTHSC, and the whole skull is sent across to Duke. And that means the brain's still in the skull. There's no distortion. So if you're taking the brain out of the skull, you could damage it, you could distort it. It's just going to sort of move because it hasn't got that protection and sort of the skull. And what you can do with this is not just sort of volume, but also various connectivity measures. So these are some images from a paper we published last year. Where
David, this is Andy again. I just have a technical question because I've been in that business of trying to scan animals. And so when you take the heads, you're fixing them, right? Are you like,
yes.
So you're doing an intracardiac perfusion or something to make sure the
Exactly.
vein is really well preserved?
Yes, exactly.
Okay, got it. Okay, thanks.
So yes, they're perfused so straight away after being euthanized. And again, that's something John and Jesse and Melinda have been doing. So yes, this is a paper we published last year where we just looked at 4 strains. So C57 Black 6J, DBA2J, because they're the parents of the BXD, Castaneus, because it's a different substrain of mouse, and BTBR, because it has a known cortical defect. And hopefully you can see that all of these have a nice connection between the 2 hemispheres, and it's missing in BTBR. So that's a nice thing. We can see something we thought we were going to see. But 2 things I really like about these images are these are 4 males, sort of, each of these is made up of 4 males clapped together. Each of these is made up of 4 females clapped together. And you can see differences in shape of brain regions that are really similar between the same strains. So you get these sort of weird things in the olfactory bulb that are similar between males and females, but are different between the strains. So this is telling you, you can see really small differences between strains, genetic differences, and there's not much difference between the sexes. And again, just to sort of show that I've not just cherry picked one particular region showing that, this is another different slice further down. And again, you can see the similarities between males and females and the differences between the strains. So as I said, we had these different measures and we had what we call these Dti measures, which are measures of sort of the movement of water molecules. And what these tell you about is white matter, fiber tracks, sort of the number of fiber tracks, their myelination. So I'm not going to go too deeply into what these are, but that's kind of what these are showing, a white matter. And then we have the volume measures. And we had 332 different regions of the brain that we sort of split up with these atlases. And we looked at heritability of all of these regions. And we split out with and without BTBR, because of BTBR was a bit of an outlier, it can increase heritability. And what you can see is pretty much every brain region we looked at has heritable differences in volume even between these 4 strains. And many regions had heritable differences in these DTI measures, these connectivity measures. So this tells us both those volumes and those scalar measures, the DTI, are highly heritable. So we tested this to make sure this was true in various ways. One way we did it was to estimate heritability in males, heritability in females, and see how well they correlated. And they correlated very well. Another way was to look at the heritability in different sizes of brain region. So you could imagine that if you have a really tiny brain region, you're going to have more noise, it'd be more difficult to measure it, and that would reduce the heritability. But we didn't see that. Even small brain regions got high heritability on. And the third thing we did to check this was to look at the 2 sides of the brain. So 2 sides of the brain are pretty symmetrical. So if you measure hippocampus volume on the left-hand side, it should be similar to hippocampus volume of the right-hand side of that same individual. And again, we see a very low coefficient of variation between the 2 regions of the brain, 2 hemispheres. So this gives us really good confidence that we're able to accurately measure many regions of the brain, both in terms of volume and connectivity. And as I said before, the vast majority of brain regions had a significant strain effect on volume. A lot of these were caused by Castaneus. So this is volume and all sort of significant strain by strain interactions. But even between B6 and D2, which are 2 sort of classical inbred strains, 175 brain regions had significant differences between the 2. And at least all of those will segregate in the BXD family. And as we saw before, even things that don't segregate between the parents can segregate in the family. So this tells us that, yes, the BXD will be an excellent resource to look at brain differences using this MRI technique. What we also see is not much difference in sex effects. So we saw that in the images. When we do the analysis, we see the same thing. But those regions that we do see a sex effect on seem real. So the most significant region with a sex difference with the bed nucleus of the striatum terminalis, and that is known to have sexual dimorphism in humans as well. So it's not that we can't detect sex effects, it's that they're not there in the mouse. And the final sort of static image I'm going to show you is these connectivity measures. So what is basically done here is you take one of sort of 4 regions of the brain and ask what regions are connected with it. And I didn't explain the colors before. I told you I'd forget to say something. So if we go back up here, front to back is colored green. So this is those DTI measures. Fiber tracks moving front to back are in green. Those moving left to right are in red. And those on the Z-axis sort of towards us and away from us is in blue. And that's the same here. And again, you have the 4 strains, B6, BTBR, cutaneous, D2, and you can see the clear hemispheric deficit in BTBR and the differences in connectivity between the strains which connect to the same area. So we have a whole brain, and in fact, it's not just the whole brain, it's the whole head. So we can look at eyes and ear structure if we wanted to, but in terms of Alzheimer's disease, we're going to talk about the whole brain. So you're not having to pick and choose, oh, I'm going to dissect out the hippocampus. You can look at the whole brain in situ. Because it's still in the skull, you have very little or no distortion. And this means that you can look at the hippocampus, for example, in this strain and in this individual and in this individual, and say we really are measuring the same thing. So it's quantitative and it's alignable between these different genomes. And therefore it's something you can use for all of your analysis. And again, we're looking at sort of all fiber tracks. We're not having to sort of narrow down what we're looking at. What we're then combining this with, Andy, do you want to say something? Sorry.
Yeah, I had a question, David, a couple questions. So basically that colorization that we were looking at that's so beautiful, that's not really volumes we're looking at. Those are tracts.
Yes, this is fiber tracts.
DTI. Okay. Yeah. And my other question was, you know, you mentioned you can look at any kind of like system you want, but you're looking at the whole brain. But I have to ask you, and I know I've talked to Rob about this, you know, given that your risk of Alzheimer's in a person goes up like, I don't know, 7-fold if you have sensory neural hearing loss in middle age, and it goes up, I forget if it's like 3 or 5-fold if you have loss of olfaction. To me, it is so tempting to want to drill down on those areas in your model. And I'm just curious, I'm sure you're aware of that information because it's out there in the lay press, but curious what parts of your analysis will include some measures of, you know, olfactory bulb size or something like that.
Yeah, we'll start off being sort of agnostic and sort of look at most everything. Again, another advantage of the BXD, we have measures of sort of hearing loss, or vision loss and things like that. So we can see if any of those correlate with both the AD measures and the brain measures. So again, we're not just doing this for the AD-BXD in the transgenics, we're also doing the matched controls. So we'll be able to see what's happening over time and say, hey, this strain has hearing loss. And we see the brain sort of correlates to that. And then that causes worse Alzheimer's disease phenotypes, or the matched AD actually just has the same kind of thing as we'd expect as the non-transgenic of the same strain. Yeah.
Thanks.
No problem. So now what we can layer on top of that MRI is light sheet. So now the same brain we ship up to Life Canvas, and they take it out of the skull, and there's potential for damage and distortion at that stage. They use this clearing technique to basically make the brain see-through, more potential of distortion. We know that distortion isn't necessarily equal, it can be non-predictable, it can be different in different directions. And then they can put in antibodies that we can visualize. what is beautiful about using the same brain is that we can map the light sheet back to that really good MRI image we have. We can take points of the brain, for example, the ventricles, and sort of distort that brain back to what it was before. So again, Alan and his team have done great work on that, sort of on the bioinformatics side. And in this light sheet, we can use sort of any labels we want, any sort of antibodies. We can use endogenous fluorochromes, so transgenic animals. We can get basically a cell level resolution on the light sheet. And again, we can get any sort of measures you want. You could look at where are dopamine receptors expressed, where are activated microglia. And so I'm going to show you some more nice videos. So this is a yellow fluorescent protein driven by the Thy1 promoter in neurons. And this is the light sheet to map back to an MRI. And you can see neurons, and you can see sort of different regions of the brain. And let's see what. So, and you can see sort of that you have this almost sort of cell level resolution. And you can do this, as I said, with sort of any of the many transgenics that are out there, or you could do this with an antibody. And I'm trying to kill time for a second because I really love this fly through that it's about to do. So you get this fly through where you can basically see single cell resolution as you move through the brain. But that's just the light sheet. Because it's the same brains, we can map back our other measures from the MRI. So we can map the tracts back onto that same brain. So this is the green for the Thy1, and we can see the white matter tracts connecting these up. And now you can imagine having this across 30, 40, 50 BXD strains at different ages, the two sexes, and having all the phenotypic measures that we can correlate with it. So I think these are absolutely sort of gorgeous images. And this is all work that is ongoing, that people are working hard on at the moment. So the final thing I'm going to talk to you about is what next? What haven't we done? And one thing we haven't done is increasing the phenotypic complexity. We take snapshots of behavior. We say this particular day at approximately 6 months old, and this particular day approximately 14 months old. And that's kind of all we know about the behavior of that animal. What we want to be able to do is monitor the animal for months, years, and see how its behavior changes over time, both on the short term of sort of circadian differences and on the long term of exactly what age does the cognitive decline start. And can we map that, for example, maybe sort of something at 14 months was actually really good up until 13 and a half months and then went off a cliff, and another strain was actually fairly terrible at 9 months. And we wouldn't know that with just these snapshots. And one way we can get at that is using these colony racks. So this is something we're really interested in. And what effectively these are, they're your normal mouse room racks with 70 cages in, but these cages are connected up in sort of any sort of way you want them to be. Each of the mice has an RFID tag in it. So the same thing you've got in your pet dog or your pet cat, just in the back of the mouse's neck. And each of the tubes has an RFID receiver. So you know when the mouse goes into the tube, when it goes into the cage, when it comes out of it. And because these are just your sort of normal housing racks, your mouse can stay in there for as long as possible. You could have a mouse that was born in there and dies in there, and you get the entire lifespan. And this is work that Rupert Overall, Sarah Zoka, and York Winter are doing in Germany. And what they can do is, so these are sort of the racks with the cages or without the cages, and you can see that the different ways they're connected up. They can look at movement, so you can look at a particular mouse, how much is it moving around? You can look at space usage. Do they have a favorite cage that they always go back to? You can track friends, so which mice prefer to be together. And you can get all these sort of naturalistic behaviors. We can map indirect genetic effects. So if you put individuals of several different strains in there, you can map what is due to their own genome and what is due to the genome of those around them. So if you've got an aggressive strain in there, maybe you spend your time trying to avoid them. But all of those naturalistic measures, really interesting, but they're not telling us the same thing as our cognitive tests. What we want to be able to do is ask the same sort of questions, but the mouse can now sort of do it whenever they want to in sort of a natural way. So we could do things like put running wheels in there and see which mice prefer to go for a run. We can put sort of choice tests or learning tasks in there. So you could imagine having a cage in there that has a light that can come on and off. And if the mouse has to learn that it pokes its head through to get a tasty treat when the light is on. You can have sort of risk reward anxiety tasks. So you could have a tasty treat in one cage, but they have to get through a cage that smells of fox urine or something like that they're scared of. And we can add cameras onto it. So not just use that FRID, but sort of put cameras on, and then you can look at grooming behavior, both grooming themselves or grooming others. You can look at gait. So that's sort of another measure of physical decline, is how does their gait change over time. So these are things we'd like to be able to do because we have sort of genetic complexity and phenotypic complexity, but we don't have that sort of time frame aspect, and that's what we want to add in next. So I'll conclude and say, please embrace complexity at all levels. We have all sorts of new systems and new technology that allows you to do this, and at scale. We have the Diallel cross to add genetic complexity, because every time you do something in a single genome type, you risk both false positives and false negatives. We have tools for deep phenotyping. You don't have to worry that, oh no, I've got 200 hours of videos, I'm never going to find a student who's going to look through them all. You can train a machine learning model and it'll look through them all and give you sort of quantitative differences. We have these new brain imaging techniques that allow us to look at many individuals in sort of unprecedented depth, and we can now sort of use new tools to add in sort of time. So thank you all for listening to me. Thank you to everyone here at UTHSC. So Rob's driven a lot of this. Casey, Melinda, and Jesse have done a lot of the work with the BXD and maintaining the breeding colonies. John has collected all this behavioral data and done a heck of a lot of work on that. Thank you to everyone at Duke in Al Johnson's lab, because they're the ones that do the magic of turning this sort of mouse brain into those beautiful images and then quantitative values that we can actually map. Thank you to Catherine for sort of developing the ADBXD model and giving us support. And also Marcelo, who I've not talked about, but he's starting to do some functional MRI in these ADBXD. And also thank you to NIA, who are paying for this as well. And I'm happy to take any questions.
David, I've got one. Something I forgot that we did in open field in Mike McDonald's lab in his Alzheimer's model was to count fecal pellets. Not that I'm looking to get any more work, but just looking, I wasn't sure quite why we were doing it. I didn't look into it, but now that I've done 250, 300 mice, I will say that one cage hardly defecates at all in open field and another one will. So there may be some strain difference between the two, showing anxiety and all that. So I don't know if that's something we might want to keep track of.
Yeah, exactly. And that's the beauty of having the videos, is that
that's true.
We don't have to have you in the lab counting the number of them that come out in each test. We can go back to the videos and have a machine learning algorithm that just says, okay, at 9 minutes and 90 seconds or whatever the sort of end of the test is, there's these many fecal boli in the arena. And so, yeah, we can pull out all those kind of measures.
And also with the new racks that I've been following the emails from Rob about the new racks, and but would we do multiple sexes, male and female, in that same, and just pull out the females when they're pregnant, or would keep it all different strains and all males or all females, or how do we propose working that?
So the easiest way to do it is all females, because they fight less. So that's what we've sent them over to Rupert, and that's what he's done. We were chatting about ways to put males in there, whether you could have, so you could put gates on them, so that if an RFID tag is red, it'll close the gate. So you could imagine having 6 males in there that are all sort of closed into their own little arenas, and the females get to choose which males they go and visit. So we could do all kinds of experiments with that. But so the initial idea would be do all females, because then you haven't got the problems of pregnancy and males fighting and that kind of thing.
Yeah. I mean, when I initially thought that, I thought we were going to put our current DXP mice in these and said, oh, I'll never find my mice,
Yes. Yeah. The advantage is you could get a readout of, okay, I know that RFID went into that cage. I can pull that cage out. But yes, this is sort of a different experiment.
And we used RFID tags in McDonald's lab, so I'm pretty good at putting those in.
Yeah, and those same sort of things we've got on the cameras now, rather than sort of scanning a tag that we're doing at the moment, you'd just be able to scan the mouse and it should label all the videos with, that's this mouse.
David, can you look at the chat questions? There's one from...
there's 11 of them. That's exciting.
Yeah, the one from Genji, I don't think has been covered at 3:50. Jen, you want to ask your question?
Yes. So I was wondering, like, so is that the Diallel cross test can be used to like help find the genes that underpinning the difference of different phenotypes? So I was wondering, does it mean that you can use that to like finally help finding those genes that might contribute to the phenotypes that you observe in the brain, and probably some genes that relate to the incidence of Alzheimer's disease?
Yes, exactly. That's exactly the plan. Because you have the genetically variable BXDs, we can associate regions of the genome. Being honest, it might be a stretch to say genes will probably have sort of a handful of candidate genes within each QTL, but you can say that this region on, so let me share my screen again. We could say, so go forwards, we can say that this region on chromosome 12 contributes to amyloid beta in our ADBXD cross. And we can say this region on chromosome 4 contributes to the differences in short-term memory in our ADBXD cross. So that's exactly right. We can associate genes with differences in phenotype in these transgenics.
This sounds amazing. Thank you.
I think it is, but I'm biased. Let's see what else we've got in the chat.
There's a poop discussion.
Excellent. Yeah, we might even be able to do that, because if they're different sizes and different shapes, we can teach a machine learning model to learn about different.
I'm proud to say I'm a professional on these things.
Kate and Megan have this great assay they developed with Amelie Beau, where you just pick the mouse up and see to what extent it pees. And that would be an easy one for John to add. Not that John needs any more work, but it is an easy assay.
We could even do that in the videos though, because the puddles are fairly easy to see, and you could.
Well, and if he has to read ear tags prior to putting them in the field, then that's all it is. It's the same amount of work. It's just marking like a dot on the sheet.
I had a question about the machine learning. So is it Simba or Deep Lab Cut that incorporate the maze, or is the maze not incorporated?
So what I've done is Deep Lab Cut basically takes the video. It can know where the corners of the maze are. So I basically taught it where the corners are, and then what it basically outputs is a great big table saying the nose is this distance away from the center of the mouse and this distance away from each of the corner of the maze. And I then input that into R, and R sort of says, okay, this part of the maze is this part of the maze is this, and this part of the mouse is in that part of the maze. So the analysis on that side is being done in R. And that's all just Deep Lab Cut and R. Simba sort of thing is doing more, would be looking at more complicated behaviors. So sort of differentiating grooming from just sort of sitting still, for example.
And right, okay, so like speed and like mobility, immobility, you could either get from deep lab cut in R or train Simba.
Yeah, I pulled that out straight away with deep lab cut in R. You get sort of the amount they move, their overall sort of raw speed, and then the speed when they're moving.
Cool. And I presume you're only doing one mouse at a time?
Yes, yep. We've not added in the complication of doing indirect genetic effects or anything like that.
I mean, we're doing simultaneous 2 mazes at a time, but that's it. So, but I do have another question. So in the beginning, we had mice jumping out of the Y in the snowflake maze, and then we elevated them. So we're kind of doing elevated Y maze. But so I'm having fewer mice jump out, but how does that affect? I mean, we just has left. It's just
another phenotype.
Yeah.
So yeah, that's a problem that we sort of had to try and get around. So hopefully we've solved that. But yeah, that's kind of a problem. It will tell me when they don't and sort of throw it up as an error. But yeah, that's kind of, it's problematic.
I
Or
think
you
some
can just
people,
make the wall taller.
well,
Yes.
so, yeah, usually like some people cap it, like have a ceiling on the Y maze, so they're enclosed in there.
Yeah, there's one that we have where there's a little door and you can put them in there, and maybe that's something to think about.
So I started a few months ago, or a few cycles ago, recording when I weigh the aging colony every 2 months, which mice try to jump out of the weigh boat. So that might, I'm trying to get some data on that. That might be interesting.
Yeah, and again, that sort of, is it activity, is it anxiety? So yeah, it's these kind of various phenotypes that probably correlate with each other a bit.
Wait,
But
and
just-
age.
Yeah.
But just as the mice get on top of the Lego figures, sometimes they like to go up on the edge of and look around and walk on the edge. So that's a whole nother thing where they're walking on the edge of the maze and not just jumping out.
Yeah, that at least isn't a problem for sort of the Y maze, because they're still going into the different bits, so they're still doing the basic test.
It's
So David,
interesting in a phenotype way though, because I find that too. Some mice will jump on the edge of the weigh boat and just kind of balance there, and others will jump completely out.
So David, great talk. Couple of questions. One is, one comment on the poop thing. It's really easy to just write any off-the-shelf object recognition algorithm to detect poop. We've done that, and it's easy. So another comment is on a question on your what where which test. Are there any signs on the wall to tell the mice which one is left and which one is right? How do they know which one's left, which one's right? Because it looks like your 2 objects are symmetrical, right?
Yeah, more or less symmetrical. They're in the same place. So you have sort of the rest of the room. You also have the camera across the top. So one side of it, they'll be able to look up and see the sort of the camera there. And in the second contest, the sort of stripes aren't symmetrical on the walls. I made it look like they were sort of nicely striped. So in the second context, they should be able to tell left and right because of the stripes as well.
I see.
But the stripes are on all 4 walls.
Yes, but they're not symmetrical. So they can sort of see sort of, okay, that side has, yeah.
So a question on genetics, which is, have you thought about some kind of formal way to test epistasis or gene-gene interactions within this context of the Diallel cross?
So the Diallel cross with a transgene or real sort of epistasis in sort of
Not with the transgene.
Not with the transgene. So yes, there is sort of the sort of 2 scan that RQTL can do, which is sort of an epistatic effect. I've not really thought about it with Diallel cross simply because we haven't got that amount of data. We need sort of think about getting that data and how we'd analyze it. We can do, as I say, that sort of 2 scan, but it'd be really nice to have a sort of X scan of how many different regions of the genome work together.
It's not really 2, right? It can be
N,
50.
whatever N is, yes, exactly. That's what we'd love to be able to have, but that's the power problem. Even if you're using all 140 BXDs, you just don't have the power to see that sort of epistasis past 2. Once we have Diallel cross with potentially thousands of individuals, you should have then the power to see it, and we need the tools to do it. So,
but then you have to do. I mean, then you still have a practical problem of phenotyping all of them. And
yes.
Is there a way you can sort of know which strain you need to pick to do the phenotyping ahead of the?
So yeah, you can do that a bit with. So this comes back to the prediction side of things, because we know so much about the BXDs. You could say, okay, I'm going to cross short-lived to short-lived, long-lived to long-lived, a really short to a really long, and say, I expect these to be sort of at the extremes, do the classics of sort of Thylal crosses of, if we expect it to be additive, we'll take a high one and a low one, and it should be somewhere in the middle, or maybe it's going to turn out to be dominant or something like that. And we can do the reciprocals and see if there's some sort of in utero effect, for example. So in the short answer to your question, yes, you can pick out the extremes of the parents to use, and hopefully that will give you sort of extremes in the F1s as well.
Yeah, there's some work by William Valdar. I think he looked at epistasis in a Diallel cross pretty carefully. And we do have one data set that was used by a collaborator in Shenzhen, where Shusheng came from, that actually did 3-way epistasis in a small Diallel we generated of 76 CXBs crossed every which way.
Cool.
Yeah, I thought
David?
Yes, go ahead.
I have a question. So you said that you did MRI, right? MRI of brain.
Yes.
So where is that MRI at UT where you could scan brain?
So this is at Duke. So we send the whole brain, a whole head across to Duke University, Durham, and they do all the imaging.
Makes sense. Next question I have is, okay. Next question I have is, have you ever stained this brain, like a full brain where like you have like a cerebrum, cerebellum, and olfactory bulb? The reason I am telling you is I tried to stain. I was able to stain, but the problem happened is I could not find any microscope that could give me the whole brain image. And the second challenge I faced is like I wanted the dorsal section or sagittal section for each, like cerebrum, cerebellum, and olfactory bulb, and I could not get it. So how have you ever done it, and any experience on that you could suggest me?
I don't have much experience with that. What we're doing is the whole brain stays solid. So we've got MRI and this cleared light sheet. So we're not sectioning the brain. The light sheet is sort of while the brain's still intact. So we can take any plane you want and take any region. So yes, we could sort of zoom in on some really small region that you're interested in and get you the whole plane in both directions. So that's kind of those videos I was showing you, because we have kind of every plane and every angle, we can make that kind of video of the whole brain or any particular brain region. I say we can, what I mean is Al and his team do some magic to do that I don't really understand too well.
So that is a live mice then, right?
No, dead mice. So just
dead mice. Okay.
Yes.
So that is also not at UT, you are saying?
Yes, no, that's over at Duke.
Okay, so that is Duke University, you said?
Yes, yes.
Okay, thank you. Another question about your genetic background. Like I have one question about genetic marker. For how do you verify your genetic background enrichment using those marker? And because I asked Jesse that do you have any like PCR or something that I can do it? So I was not able to get the answer properly. So how do you get the enrichment of your genetic background? For example, I have just say C57BL6J, and that I crossed to S129V, and then I kept back crossing few times, and I wanted to know what is the percentage of C57BL6J and S129V.
So you could do it two ways. You could just sequence the individual and you'll have sort of mark, because the two parents have already been deeply sequenced by others. You can just say how many regions of the genome map back to the two parents. There's also the sort of Muga arrays and mini Muga arrays that have a lot of known markers for these inbred strains. That's very cheap, and it's what sort of JACs use to keep track of their breeding. And effectively, it's just you take DNA, you put it on the mini Muga, and it'll say how many variants on there are from Black 6J, how many are from DBA, or whatever sort of the many sort of inbred strains that are designed to have that. Because there is, and also it will depend on how many generations you've done. If it's an F1, we can sort of, denote in silico in advance what the genome is going to be. In an N2, you're only going to need a very small number of markers because it's only one or two recombinations per genome. If you're getting into more generations, you'll need more markers. But no, sort of that mini Muga is probably the cheapest and quickest way of doing it, and it's sort of one of the classic ways that's done. It's done now.
It's $40. I put some links in the chat for you.
Yes, Megan is probably the better person to ask because she.
That is again from Jackson Lab, right?
So no, so
Sorry?
you can order this and sort of use it. What I was saying is JACs use this to keep track of their breeding.
Yes, because I had contacted Jackson Lab and they told me the same thing, send the DNA and then we will sequence them. But just I was wondering in GZI if we have any facility that we can do quickly.
Yes, those links that Megan sent and sort of, I think Megan's probably the, I'm sorry to throw something onto Megan, but I think she's probably the best person to ask because she's done a lot of this kind of thing.
Yep. So
Okay.
Roshan, just go to the first floor of the same building you're in every day and go into the MRC and look for the tag center and just put a tail in there and they might charge you a couple $100, but they'll give you the answer. More than that, less than that?
It's
Okay.
it's $10 to extract the DNA from the tail and $40 to get the mini Muga.
Perfect.
Cheap, very cheap.
So what, about $50 per mouse?
Yeah. So I just wanted to comment that I thought this was a great presentation, very lucid. What's impressive is that David gave primarily a technology and resource talk, but he could have given 2 or 3 science, kind of standard science talks. And I thank you personally for doing the resources, because I think the cool thing about a resource talk is everybody and their mother can use these resources. They're here now uniquely at UTHSC. There's no place in the world that really does this kind of work better than we do. And so if you switch, if you're using a Black 6 129 strain model, somebody, you should immediately, if not sooner, switch to BXDs and address the problem with the BXDs, because you'll get a lot farther, faster, and you'll have a lot of collaborators in the TSRB and elsewhere who can help you with everything. And the phenotyping, I just want to point out that obviously people have been doing behavior genetics forever, but this is where, thanks to computers and cheap images, and everybody has one of these things in their pocket that is really a touch screen. So you can turn these into a monitor for mouse, you can train, they can record. So we have this amazing technology that is now going to affect behavior genetics over the next couple of decades. And it's going to be, we're going to know so much in 10 or 20 years.
Yeah, I could try to throw it in there that this can be applied to anything, any sort of those transgenic models that have been developed over the last sort of 20 years. And you've had things like the Knockout Consortium, who've sort of generated sort of 20,000 knockouts but all on the same genetic background. And so any of those could be used. And again, this sort of video-based phenotyping, you can take any of your classical measures that you've been using for the last 50 years and sort of add more to them. And also it means that you don't have to have sort of 50 undergrads in your lab that you're forcing to go through every video frame by frame. I don't think anyone enjoys doing that. You can get one person to annotate it who knows the behavior really well, and then this computer can then sort of just do any videos you do in advance. Any other questions?
You've been nominated to lead monthly machine learning pose estimation and behavior club.
That's just going to turn into me getting you to annotate videos for an hour.
That's fine. All right, well, if there's no more questions, that was a fantastic presentation, seminar, learning experience, David. I thank you. And thanks everyone for attending. Great questions. Hopefully we'll have more discussion about development of these resources.
Yeah, please, anyone, send me an email, talk to me. Anyone's interested in using these, anyone's interested in analyzing any of these. We have a huge amount of data. John is generating a huge amount of videos. So the more the merrier.
Awesome. All right, everyone. Have a great weekend.
You too.